Dear students, now we know that there are so many types of RNAs and they perform a vast variety of functions within the biological system. And the fact that all of these functions are a result of the RNA structure. But one wonders, why do RNAs form the structure? What is the advantage of an RNA molecule to fold onto itself and take form of a structure? So to motivate you towards this topic, underlying theme is the stability of the RNA molecule. If an RNA molecule becomes more stable after folding, then it is going to fold and of course take form of a structure. So once they become stable, they can start performing their function within the biological systems. However, can an RNA molecule take multiple forms in terms of their structure? The answer to this question is yes. And it's fascinating to see how the same RNA, single-stranded RNA molecule, can take form of the multiple structures. So in this module, we will see how a single-stranded RNA with the same nucleotide sequence, remember the nucleotides that are forming the RNA, how the same sequence can result in multiple structures and what is the underlying theory of forming these multiple structures. So for that, you will have to look at the concept of free energy, specifically the Gibbs free energy. As you can see on the slide, it is the free energy of an RNA molecule that is available for reaction. So obviously, if there is more energy available within an RNA molecule to react, then it is unstable and it wants to react to form a compound that is the result of the reaction. So if an RNA molecule has a very low reaction energy or very low Gibbs energy, then it means it is more stable. As I said, smaller Gibbs free energy, the better. And if this results as the formation, after the formation of a structure, all the better, because one, the molecule is more stable, and two, the molecule has taken up form of a structure that can perform some very important functions within the biological systems. So to summarize, the RNA structure formation, the process, it lowers the free energy that is available within the RNA molecule. So RNA structures have the lowest or close to the lowest amount of free energy. An interesting question can be, is there a structure that exists in nature which has the free energy greater than some other RNA structure obtained from the same nucleotide sequence, which has an even lower free energy? I leave that question for you to think about. In case of theoretical studies and bioinformatics, what we do is we try to form structures out of the nucleotide sequences of RNA. So if there are, let's say, 5 structures that can be obtained from the same nucleotide sequence, we simply select the lowest one, the one with the lowest free energy, because it's the most stable. Here, I'm showing you an example of how complementary base pairs, they come together with hydrogen bonds and force the RNA backbone to take formation of a structure. As you can see here, C is paired with G, and after that, G is paired with a C, and following that, A is paired with a U. So it's like two people holding the different ends of the RNA molecule are coming together, leading to the formation of a secondary structure. As you can see, in A and B, two exactly same structures can be formed as the result of this process. However, the underlying nucleotide sequence is quite different between A and B, as you can see in this diagram. Okay, so moving on, how do you calculate the energy of an RNA structure? For that, you have to divide, you have to segregate the good energy, the stability energy, and the bad energy, right? So essentially, energy is a scalar, and there is no good and bad. But if the RNA nucleotides, they come together and form a hydrogen bond, some energy is given out. So we call this the good energy with a negative value. However, as a result of folding, if there is some instability that is introduced within the structure, we call it the positive energy. However, it adds towards the destabilization of the RNA molecule. So to look at an RNA structure, all you have to do is look at the negative energies and the positive energies, sum them up, and if you arrive at a stable molecule, then that's your structure. However, if you want to see how the structures are formed and how the energy is released or stored within the structure, you have to see which nucleotide, if coupled with which other nucleotide, gives out how much energy, right? So you may want to couple those nucleotides, complementary nucleotides, that give out the most energy. So for that, you can look at this table in which you can see that if an A is coupled with a U, then minus 2.1 kilocalories per mole is given out, right? Similarly, if U is coupled with an A, then another minus 2.1 kilocalories is given out. So by looking at this table, you can look up the energy that is given out as a result of coupling of the two nucleotides. Right. So there is some food for thought at the end of this module. So this is just the stabilization energy, right? The energy that is given out as a result of coupling complementary nucleotides. But where is the positive energy that I was talking about? So this was the destabilization energy. How can we measure that energy? Where does it come from? So all of these questions are for you to think about. And in order to list down the destabilization energies, you have to factor in the things that underline destabilization of the RNA.